Hi everyone, we are starting a new chapter, Genetics of ICSE Class 10. This is a very important chapter and some of the important questions have been selected. The first question is, name the following. The ratio of the offsprings in F2 generation in a dihybrid cross. Now, in order to derive the ratio, we should understand what a dihybrid cross is. We know that Mendel had discovered two crosses, monohybrid cross and the dihybrid cross. Dihybrid cross is a cross in which parents are taken which differ in two contrasting characters. That means when two contrasting characters are considered at a time, it is called a dihybrid cross. For example, when round yellow seeds are crossed with wrinkled green seeds, the resulting progeny in case of Mendel's experiment were all round in yellow. This was the F1 generation. Now, on self-pollination of the F1 generation, what we got the ratio 9:3:3:1, where round yellow were 9, wrinkled green were just 1, whereas we had two new recombinants as in round seed with green color and wrinkled with yellow. So these were the new recombinants which were formed and they were 3 and 3. Hence, the ratio is 9:3:3:1 in case of a dihybrid cross. The second is the fundamental unit of heredity. We know that genes are the fundamental unit of heredity. So before we talk about genes, we should know that where exactly the genetic material of an organism is. We know that the genetic material of an organism is inside the nucleus. So when a cell is not dividing, it is in the form of chromatin. And at the time of cell division, the chromatin material condenses to form shortened thick chromosomes. And the chromosomes basically contain DNA. Segments of DNA which can make a protein or a character are called genes. So human beings have got many genes which are responsible for all the characters which we see. It could be eye color, height, intelligence, etc. Next, the term used when a cell has more than 2 sets of chromosomes. Now, we know that we have different kinds of cells in our body. We have haploid, we have diploid, we have polyploid. So what do you mean by a haploid cell? When we have one set of chromosomes, then it is called a haploid cell. For example, all the gametes are haploid. When we have 2 sets of chromosomes, that is one from the parents, one from the maternal side and the other from the paternal side, it is diploid. When organisms have more than 2 sets, then they are called polyploids. For example, banana is triploid, some varieties of wheat are tetraploids, hexaploids, etc. So polyploids basically means that organisms having more than 2 sets of chromosomes. Next, the term used for mating of closely related individuals. This means that when organisms or animals or plants of the same breed are involved in mating, they are known as, or this phenomena is known as inbreeding. Now, what is the purpose of inbreeding? Inbreeding is basically done to incorporate the superior characteristics. So superior male and superior female are selected and then they are mated of the same species, obviously. They are mated so that the resultant progeny has got all the superior characteristics. And the next one is a pair of chromosomes carrying dissimilar alleles for a particular character. Now we know that Mendel had used certain terminologies in genetics like heterozygous, homozygous. So what does heterozygous mean? What do you understand by the term heterozygous? We know that each gene has got 2 alleles. When both the alleles are different, then such chromosomes carrying dissimilar alleles for a particular character are called heterozygous. For example, if tallness is a character, then when it is denoted by a capital T and a small t, and so that is the individual members are different in the gene, then such chromosomes are called heterozygous.